Thank you very much for joining this seminar today, part of the series on combining multiple genomics methods to study complex disease. I'm delighted to introduce today's speaker, Professor Connie Bezzina. Connie is Professor of Genetics of Cardiac Disorder at the Academic Medical Center in Amsterdam and will present data from her laboratory and ongoing collaborative studies from the International Brugada Syndrome Genetics Consortium.
Thank you very much for that kind introduction. Good afternoon to everybody on this side of the world, and good morning to everybody in other parts of the world. So the title of my talk is Dissecting the Genetic Basis of Brugada Syndrome, and I will be sharing with you in the coming few minutes to 1 hour our recent work and our planned and ongoing work concerning genetic studies in Brugada syndrome. Before going to the genetics, I would like to share with you some numbers concerning sudden cardiac death, which forms the basis of why our group is focusing on research on the genetic basis of sudden cardiac death. So roughly 1 out of every 1,000 individuals per year suffer sudden cardiac death, and as such, this accounts for roughly 20% of total mortality in the general population. In the United States alone, sudden cardiac death accounts for approximately 400,000 cases per year. Furthermore, if we consider death or mortality from cardiovascular diseases, we see that half of individuals that die of a cardiovascular cause die suddenly and unexpected. This means that sudden cardiac death is an important societal burden. What we see here is a normal cardiac rhythm. In the setting of a normal cardiac rhythm, the electrical impulse is generated in the sinoatrial node, which you can see this little place up here on the left-hand corner, and the electrical impulse is then passed in a very coordinated fashion throughout the heart. What this leads to is a very coordinated contraction of the ventricular chambers of the heart, which leads to efficient pumping of blood throughout the body. In contrast, this is the heart beating, or rather not beating at all, in the setting of ventricular fibrillation. In ventricular fibrillation, which is the most common arrhythmia that underlies sudden cardiac death, the electrical function of the heart is now not normal, it is very chaotic, and as such, because of this, the heart, instead of contracting in a coordinated manner, it instead quivers uselessly. This leads to circulatory collapse, and critically, if this arrhythmia is not stopped within minutes, death will ensue. Now, so arrhythmia can be stopped by a defibrillatory shock, by an external automated defibrillator, or by an implantable cardioverter defibrillator, but unfortunately, in the great majority of cases, help to stop arrhythmia comes very late, comes too late. And this underlies the large mortality that we see due to this particular arrhythmia. Now, because of that, this means that prevention is key to solving the problem of sudden cardiac death. Prevention is possible, as I said, by means of an implantation of an ICD device or also by drug therapy, but this emphasizes the point that we need means to identify those individuals in the general population that are at risk for dying suddenly, such that we can handle and treat them preventively. Now, sudden cardiac death occurs in the different age spectra of the population. It affects the old, it unfortunately also affects the young, and in the different age strata, there are different cardiac diseases that are associated with risk for sudden cardiac death. In this particular presentation, I will be talking about disorders that lead to sudden cardiac death in the young, that is, roughly individuals below 40 years of age. Now, classically, cardiac pathologies that are associated with sudden cardiac death in the young have been classified into two main categories. we have the primary electrical diseases, and so we have these shown in the figure on the left. These diseases are associated with life-threatening arrhythmia in the setting of a structurally normal heart. Many times, those diseases also show on the ECG by abnormal electrocardiographic intervals. The other main category is the cardiomyopathies, and here in the cardiomyopathies, at the basis of arrhythmia is the abnormal structure of the heart. As we can see in the bottom right panel, there we can see an echocardiogram from an individual with hypertrophic cardiomyopathy. Now, shifting a little bit gears and turning to genetics, so after the sequencing of the human genome in the early 2000s, we have seen a large number of studies, including the HapMap project and sequencing studies such as the 1000 Genomes project, the EXACT project, and more recently other such projects have spawned. These projects have taught us a great deal about genetic variation in the general population. And these studies have been made possible by the rapid decline that we have witnessed in the course of sequencing the human genome. So this is a commonly used slide which shows the tumbling of costs of sequencing the whole genome, and also showing beautifully how it has by far outpaced Moore's law. So in the previous slide, I just said that in the last 15 years, there have been a lot of studies that have taught us a lot about genetic variation in the general population, and that these studies have been largely fueled by the decline in costs in sequencing the human genome. And I was saying that this slide, which is commonly used, beautifully shows how the declining costs of sequencing the human genome have by far outpaced Moore's law. So what these studies have taught us is that genetic variation in the general population differs by virtue of differences in frequency. There are common variants, and there's a whole frequency and a whole scala and frequency of variants, from common to very rare variants, and also these are associated with different effect sizes, where we've learned that common variants are mostly associated with small effect sizes, whereas rare and very rare variants are associated with large effect sizes. And of course, this matches with what one would expect of the purifying selection on rare variants in the general population. So variants with large effect sizes are expected to be rare. In the last 15 to 20 years, the cardiac community has done a very good job in tracking down the rare and very rare alleles that underlie the inherited cardiac disorders that are associated with sudden cardiac death. And the methodology that has led to this discovery has been classical linkage analysis in large pedigrees, where first one would track down the chromosomal segment that segregates with the disease in affected families, and then by a positional approach identify candidate genes within the interval and ultimately tracking the causal mutation. So basically, these rare variants causing these Mendelian monogenic disorders would have a large effect in determination of risk for these cardiac diseases, and are expected to determine disease susceptibility for a very large part. These studies have taught us that the primary electrical diseases are largely disorders that are attributed to mutations in genes that encode for ion channels that underlie electrical function in the heart, and some of these are represented here. So ion channels are integral membrane proteins, and they underlie electrical activity and excitation contraction coupling within the heart. So the primary electrical diseases are associated by mutations in these ion channel genes, whereas we've learned that the cardiomyopathies are for a large part caused by rare genetic variants in genes encoding for structural or contractile proteins of the cardiomyocyte. These discoveries have had a huge impact in the clinical care of patients with these disorders, and the relevant professional societies such as the Heart Rhythm Society and the European Heart Rhythm Society have in recent years issued consensus statements on the appropriate use of genetic testing for these genes in these disorders. So this slide is simply to tell you that these discoveries have had a big impact on patient care, and genetic testing is now forming an integral part of the management of these patients. Having said that, there are still outstanding challenges in our understanding of the genetic basis of some of these disorders, and these challenges still hinder optimal use of genetic testing in patients and ultimately hinders patient care. So the first challenge that I will dwell on is the large variability in disease manifestations that we observe among mutation carriers. So one can imagine that, of course, these disorders are genetically very heterogeneous, so there are different genes that cause these disorders, and also for within each gene, of course, there are many different mutations that cause these disorders. So one could argue that differences in causal gene or differences in exact mutation type could underlie this variability. And for sure it does. And there are many studies out there that show that, for instance, mutation in specific parts of an ion channel are more deleterious than mutations in other parts of ion channels. However, we still observe a large variability in phenotypic severity among carriers of the same mutation. So essentially, I think that this variability can be appreciated better in large pedigrees where you have that harbor founder mutations, where you can observe the phenotype in a large number of carriers that have exactly the same causal primary genetic defect. This brings me to the next slide, which displays the phenotype associated with a founder mutation in an ion channel gene that has been identified by Paul Brink, Peter Schwartz, and co-workers in a large founder pedigree from South Africa. So this is the KCNQ1 A351V variant. As I said, it's a founder mutation. It is associated with the long QT syndrome, which is a primary electrical disorder, which besides sudden cardiac death is also associated with QT interval prolongation. So the QT interval is this interval over here on the ECG, and we know that the more the QT interval is prolonged, the more the chance of potentially life-threatening arrhythmias is increased. So there is a relation between the extent of QT interval prolongation and the chance of suffering a life-threatening cardiac event. So what we see here plotted is the QT interval for mutation carriers and non-carriers. Of course, most mutation carriers, as expected, have a QT interval that is within the normal range. But when we consider mutation carriers, we see that while most mutation carriers have a QT interval in the abnormal range, that is above the 440, 450 cutoff, there is a large degree of variability among mutation carriers. What we also see is that some mutation carriers also have QT intervals within the normal range. They seem to be protected. Now, looking at the right-hand panel and considering only mutation carriers, while we see that unfortunately most mutation carriers have suffered an arrhythmic event, there are also some mutation carriers that for some reason seem to be protected. Of course, there will be non-genetic modifiers of disease severity, and also for the long QT syndrome in particular, but also for other cardiac disorders. these have started to be identified. The effect of age, gender, and medication use has been amply demonstrated as being an important disease modifier, but also we suspect that the inheritance of additional genetic factors alongside the primary causal familial mutation will ultimately determine who reaches the threshold for displaying the disease, and among those who reach the threshold for displaying the disease, the degree of severity that they will, of disease severity that they will display. Another outstanding challenge is that some disorders, in spite of many years of research, have remained genetically unresolved. One common feature of the disorders that have remained genetically unresolved is that they have a less pronounced familial aggregation. So although, for instance, for some disorders we do see familial clustering, we see only few related individuals that are affected. And also what we also see that many cases presenting with these disorders are sporadic. That is, they don't come with a family history behind them. So we suspect that for these latter disorders, where we see less pronounced familial aggregation or sporadic presentation, we actually suspect that, contrary to the way we've been looking at them thus far, their genetic inheritance, their inheritance is likely to be more complex and is likely to be oligogenic or maybe perhaps also polygenic. So if we were to look at some of the cardiac disorders I've been mentioning, disorders like the long QT syndrome, catecholamine-induced polymorphic ventricular tachycardia would be more towards the left of this figure. They would be more near monogenic, and there are also for sure monogenic forms presenting at very young age, whereas other disorders that have remained genetically elusive and that have predominantly a sporadic presentation, like Brugada syndrome, some forms of dilated cardiomyopathy, early repolarization syndrome, and others, are most likely having an oligogenic or even polygenic inheritance. For the latter diseases, where we suspect an oligogenic and polygenic model, we think that genetic variants that are either common or have a low frequency or rare in the general population could contribute. So I would like to go a little bit further into the clinical presentation of one of these disorders, which is the Brugada syndrome. I will not bother you too much with ECGs, but just for curiosity, on the left you have a normal ECG, on the right over here you have a Brugada syndrome ECG, and the signature feature of a Brugada ECG is this feature over here, which is ST segment elevation in the right precordial leads. And Brugada syndrome, besides this ECG feature, is associated with sudden cardiac death around the 4th decade of life. In 1998, Chen and co-workers described mutations in the SCM5A genes, so very rare genetic variants underlying Brugada syndrome. There has been a lot of functional studies that have indisputably provided evidence that mutations in SCM5A are a real cause of this disorder. And several studies, such as those by Leah Crotti published in JACC, and also by the French group of Richard Redon published in Human Molecular Genetics, have shown us that rare variants in SCM5A are found in around 20% of cases. However, the remaining 75% remains genetically unresolved. As I said, Brugada syndrome presents many times sporadically, pointing to a more complex inheritance, but also we observe very low penetrance, as low in some families as 10%, and a very large variability in disease expression in families that harbor indisputably causal mutations in SCM5A. So this further provides evidence for complex inheritance in this disorder. Now, as opposed to Mendelian inheritance, complex inheritance, oligogenic inheritance, polygenic inheritance necessitates a different study design for tracking down the underlying genetic variants. Essentially, it necessitates a case control design where we compare genetic variants between cases and controls. Ideally, we would do this in an unbiased genome-wide fashion, such that we come up with robust, unbiased, hypothesis-free evidence for causality. And in such studies, we will compare the frequency of thousands of variants spread across the genome between cases and controls, where a higher than expected incidence of a SNP, particular SNP, in the disease population will suggest association with disease. So a few years ago, we set up to do this for Brugada syndrome, and for very practical reason, and also for cost, we logically started by looking into the role of common genetic variants in the form of single nucleotide polymorphisms to look for disease susceptibility variants. So the design, as I mentioned, was a case control design comparing Brugada syndrome index cases with ethnically matched individuals from the general population. So Brugada syndrome, like all these disorders I've been talking about, is a very rare disorder. The incidence has not been exactly calculated, but is assumed to be roughly affecting 1 to 2,000 individuals, but probably it is more rare than that. So it is crucial here to collaborate internationally in order to come up with the sufficient patient numbers that are necessary for well-powered genetic studies such as these. So for this purpose, we set up the International Brugada Syndrome Genetics Consortium, which now brings together more than 2,500 big Brugada syndrome probands from roughly 20 different clinical centers worldwide. The first study that we conducted has been published in 2013, and that study was a common variant genome-wide association study where we conducted a GWAS analysis in 312 Brugada syndrome index cases versus just over 1,000 controls from the general population. This is the Manhattan plot that came out of that analysis. For those of you not accustomed to see a Manhattan plot, every point over here represents a single nucleotide polymorphism, and these are grouped in chromosomes on the x-axis, and what you see on the y-axis is the negative log 10 of the p-value, and this red line over here represents the genome-wide significant threshold, which essentially is 0.05 divided by 1 million tests, which has been determined as being, so to speak, the haplotype burden in the human genome based on HapMap data and linkage disequilibrium data from HapMap. So in this analysis, in this genome-wide association analysis, we could identify 3 common genetic variants that were associated with susceptibility to Brugada syndrome. Not surprisingly, 2 of those variants were located in the vicinity of SCN5A, which is a gene which we already knew harbored rare genetic variants that contribute to the disease. So essentially, the identification of common genetic variants at this locus tells us that the SCN5A locus harbors an allelic series of variants that differ in frequency and in effect size, but are involved in Brugada syndrome susceptibility. The other locus on chromosome 6 was a new locus where we could not recognize any known susceptibility gene or any gene that had been already implicated in cardiac electrical function. We also went on to start looking at the cumulative effect of risk alleles at the 3 different loci. So what we have here on the left-hand panel in European patients and in because I forgot to tell you that this was a discovery set. We subsequently validated these 3 loci in an independent set of patients of European descent, and subsequently also extended these findings to a case control set from Japan. So as I said, we then moved on to start looking at the cumulative effects of risk alleles at the 3 loci. And in this panel over here, you can see the distribution of risk alleles in the Brugada cases, which are the clear bars, and the control population, which are the filled bars, of European origin and of Japanese origin. And as you would expect, you see a shift to the right in that Brugada cases harbor a greater burden of risk alleles as compared to the controls. But interestingly, what you see on the right is the odds ratio per different burden of risk alleles. So what you see over here is that individuals that carry 5 or more risk alleles have an odds ratio of 21 as compared to individuals that carry 1 or less risk alleles. Of course, one needs to keep in mind that this is relative, and because the incidence of Brugada syndrome is rare, one needs to keep in mind that the absolute risk associated with these variants is still small, which also implies that there are many variants out there that determine risk that still need to be discovered. So I already told you that at this locus on chromosome 6, there was no gene that was known to be important for cardiac electrical function, but one gene that immediately struck our attention was the HEY2 gene. HEY2 encodes a basic helix loop helix transcription factor that is known to play an important role in cardiac development, and hypothesizing that it could be involved in maintaining ion channel gene homeostasis in adult life, we studied the electrophysiological characteristics of mice that are haplosufficient for this transcription factor, and I will not go into more detail today about this, but essentially we validated the role of this gene in determining in modulating ion channel function and cardiac electrical function in the adult heart, and also identify the mechanism whereby it is involved in Brugada syndrome. Actually, the point I would like to make here is that these studies are important not only in dissecting the genetic architecture and understanding how can we predict risk accurately in patients, how can we identify patients that are at risk, but also important, it is a very effective unbiased approach in giving us insight into new biology and pathobiology of disorders. At this point in time, we are currently extending the common variant association analysis to the entire set of 2,500 Brugada syndrome probants, and this analysis is currently underway. So I have been talking till now about the role of common variants and what we have been doing to try to find these common variants that determine risk, but what about the low frequency and the more rare variants? Do they play a role? So this is a project that we've started recently in a collaborative effort with the group of Richard Redon in Nantes and with Elijah Baer and Pierre Lambiase in Paris, and also more recently involving the group of Nademane in Thailand. And the approach that we're using now to track down genes that harbor low frequency and rare variants in for Brugada susceptibility, we've started a new approach entailing whole genome sequencing. So as opposed to the common variant analysis, where we have used genotyping arrays to type common variants, in tracking down low-frequency variants due to their low frequency, the whole genome sequencing approach is the approach to follow and the most cost-effective. Another thing to keep in mind is that in dealing with rare variants, these are by definition very rare in the patients and in the controls, which means that we are underpowered to study their effect one by one. So an approach that we need to follow here is as an approach where we look at functional units in the genome, such as a gene unit or a regulatory unit as a super locus. So what we do is we collapse variants within a putative functional unit, such as a gene, and then check that functional unit to see whether it harbors a burden of rare variants in the cases as compared to the controls. So basically, we look as to whether the affected gene shows statistical excess of rare, probably damaging variants in the cases compared to the controls. So this is what we have set out to do. As I said, this is a collaborative project. We are planning to sequence 300 patients, 300 Brugada syndrome probes from the Netherlands, 200 from the UK, 300 probably, I think we're at more patients by now from France, and 250 patients from Thailand. Now, because the frequency of rare variants can vary greatly between individuals of different ethnic descent, it's even more imperative now to match the controls precisely to the cases. We have access to controls from the Netherlands and the UK that are sequenced on exactly and processed exactly as our Brugada syndrome cases. So we have access to these controls, but through collaboration with Project MINE. Project MINE is a very large-scale project on amyotrophic lateral sclerosis, and these investigators have been kind enough to allow us access to these controls to be used in our comparison with the Brugada syndrome cases. As concerning French controls and Thai controls, these are being sequenced in the setting of the ongoing Brugada syndrome project. So basically, this work, which has started recently, about a year ago, entails three major steps. Of course, the main first step is the whole genome sequencing of the cases and of the necessary controls. We are doing this on a HiSeq X platform, doing this at a coverage of 30x, and major steps that are involved in the sequencing pipeline involve QC of DNA to check its suitability for whole genome sequencing, genotyping, array genotyping for purposes of sample tracking and QC, library preparation, and ultimately sequence. At the end of this process, you have a FASTQ file ready to enter the next big step of this process, which is the alignment of the sequence data to the human genome and variant calling. For this, we are following the GATK pipeline that has been set out by the Broad Institute and also using other software. So here we go from FASTQ file to BEM file once we have mapped, and then we use those BEM file for a process of variant calling, which will give us our variant call files that we can then use for our case control statistical analysis. Of course, everybody knows these files are huge, so practical things to keep in mind when setting out to do such project, besides funding, of course, is the necessity for storage, for large storage space with appropriate backup. So once we have the variant call file, a third important step is the annotation of these variants. So basically, this step will annotate where is the variant located. Is the variant located within a protein coding gene, a non-protein coding gene? Which gene? Is it located in the coding region? Is it located in the non-coding region? When located in the non-coding region, for instance, it tells us whether it's located in a putative transcription factor binding site, for instance. Also, importantly, it annotates the variants by annotating their frequency using large variant databases such as HiSeq, and also what is known about possible pathogenicity of such variants, for instance, by annotating using the ClinVar database, etc. So once all that is done, then starts the process of the statistical analysis. As I mentioned, because we're dealing with rare variants, in order to achieve sufficient statistical power, we need to pool the rare variants within predefined regions and treating these regions as a single superlocus, so to speak. So, of course, we have started with looking at gene units as units where we pool variants. We will be looking at non-protein coding candidate functional regions. I think these will be the most challenging, but here we will be informed by epigenetic data that is rapidly growing in the field, and also with an agnostic approach and a completely unbiased approach, a sliding window approach across the entire genome. Statistical tests that we're using. Statistical tests are rapidly being developed in this area, and is an active area of research in itself. I think there are two categories of such tests. One of them is the burden tests, which assume that all alleles within a given locus has one particular direction of effect. So it assumes that they're either all pathogenic or all deleterious or all protective. Another test, which is a variance component, other tests, such as the variance component test, the most popular of those, I think, is SCAT, accommodate for both directions within the test. And then there are also unified tests or hybrid tests that take both possibilities into account in order to make sure that optimal power is maintained. Furthermore, a lot of research is ongoing by leading groups looking at strategies for improving statistical power. As I mentioned before, rare variants, the more a variant is rare, the more it is likely to be associated with disease. So one strategy could be to filter based on minor allele frequency and consider only very rare alleles in the case control analysis, or alternatively upweighting the most rare alleles in the analysis. Another way another strategy for improving power could be to consider particular types of mutations. So, for instance, considering loss of function mutations as opposed to missense mutations. And another strategy is to use algorithms that can tell us something about the likely pathogenicity of variants, such as the CADD score. So I would like to end with the last slide with some very, very preliminary analysis that we've done. So this analysis is a genome scan, gene-based scan in 183 cases versus 415 controls of Dutch ancestry. And of course, we were very happy to see that SCM5A, we could find that back in this data. Of course, we would like to see new genes coming up, but clearly we're still underpowered and we're still learning of how we should work with this data. I would like to end this, first of all, by thanking you all for your attention, but also special thanks to all the collaborators in this project. As I said, working with rare disease really necessitates international collaboration, and we are very privileged to be trusted by several collaborators, several clinical collaborators and genetics collaborators at various centers with collaborating with this data. So I thank all labs and all clinical centers that have provided us with patient samples, and also groups that have provided us with controls, and a special thanks to the group of Richard Redon in Nantes, with whom we carry this project in close collaboration.